the bottleneck for scientific progress, and that's really what drives us, is how can we get like new human therapies out faster? The bottleneck is turning, scientific hypotheses into data. It seems pretty obvious to me that the frontier labs are going to win long term.
But you told me before this call that you're not selling data to AI labs.
Yeah.
And yet, if all the reasoning goes through Anthropic and whatever, then at some point you have to sell.
Well, we don't sell it to them.
And so if tomorrow Nanopore said, we're not going to sell our devices to Plasmidsaurus anymore, and we're going to compete on sequencing as a service, would that not be concerning to you?
Well, it would be in violation of our agreements with them, first of all. So I'm feeling confident on that because we do have partnerships with these companies that we work with.
But this seems to be a common mistake amongst people who develop technologies, don't then sell access to the technology as a service. Welcome to the podcast. Today's guest is Mark Budde. He's the CEO of Plasmidsaurus, a company that began by offering whole plasmid DNA sequencing for $15. You've said in interviews that it took you like many days to sequence a single plasmid. You were at Caltech, and you estimated that it cost $600 to sequence a plasmid. But then you start a company and you offer it for $15. What made you so confident that you could reduce costs by 40x and actually make money?
Well, at the time that we started the company, I had figured out how to decrease costs by that point. So the way we started it is I was planning on building a therapeutics company, and we were bottlenecked on our ability to sequence plasmids. And I'd previously, at Caltech, as part of my research, used Oxford Nanopore technology for plasmid sequencing, and I suspect I was the first person to do that because it was right when it came out. Yeah, and that was laborious. The core issue is we needed to sequence like a bazillion plasmids, literally hundreds a week, and doing that with Sanger sequencing was too painful and slow. Like the issue was scalability. So with Sanger sequencing, you had to design all these primers. You had to then analyze the data automatically. And based on what I'd previously done, I knew that rather than solving that problem with like an algorithm and automating it, it would be easier to spin up an entirely different sequencing technology. So I built that for myself. And then in the process of doing that, it became abundantly clear how you could drive down costs and improve scalability on that. And then as part of that, because things were going well, I just reached out to everyone I knew at Caltech and asked them if I could try and sequence their stuff to make sure it worked. And it all went really well. And that was before we decided to launch. And then once they started begging me for more sequencing, because I was able to sequence stuff they couldn't sequence any other way, I knew that it would be okay to launch. The question about pricing, we could have certainly demanded a higher price. But, my core thinking is always, how can we maximally accelerate the pace of scientific research? And our margin certainly could have been much, much higher. But it was clear that if deployed at a reasonable price, this would be a thing that was adopted broadly versus in special use cases, and have a dramatic impact.
But at the time that you were actually starting the company, you didn't know necessarily for sure that you could get it down to 50. Like you have to do it for not $15, you have to do it for $5, right? So you can make a profit.
Yeah, but again, we built it out for our internal servicing, and there I wasn't focused on price, right? I just wanted to scale it up for myself. And in the course of doing that, it was abundantly clear we'd be able to get to where we wanted to be on price.
And so obviously Nanopore has this advantage over things like Illumina in that you can do very long read sequencing, right? Because the DNA runs through a pore and you can measure a full DNA molecule in one experiment, which presumably makes it, better or easier to automate because you just have to load these flow cells and there's not so many steps required like with Illumina. But my question is like, so you took this risk to do this, to use this new technology to do sequencing. But presumably at the time, Nanopore was so error prone that there was no guarantee this was even going to be a viable company anyway, because there's no guarantee that this technology was going to become 99.99% accurate like it would need to be
Yeah.
if you wanted to scale up customers.
Yeah.
So in some sense, you were taking a risk that the technology would get better.
Not exactly. So when I first sequenced my first plasmid back in whatever it was, like 2016 or something like that, 2015 maybe, I remember telling somebody in my lab I was going to use Nanopore for plasmid sequencing, and we were pretty seriously looking to PacBio, but ultimately the Nanopore was the best use for us. Your earlier question was about having confidence in technology. I'm a very strong believer in just doing things. It's like, in software or anything, you want to like have an architectural system in your head about where you want to end up. And then you want to be taking like just small bets to improve things every day that are directionally aligned with where you're going. And you've got to be thinking about like, is this cutting off a path towards ultimately getting where we want to be? But it was, you could see it, that there was a path towards getting where we wanted to get with just, it's just about multiplexing and simplifying library prep and doing all these things. And you could see that none of these were intractable problems. It just required going through the motions. And every time you turn that crank, you're a little bit better than the previous time. And you can just see how that is going to compound over time.
So I know famously, Plasmidsaurus did not raise much venture funding. You raised one check from a single VC I think you said just to be in that ecosystem.
We raised a small check from 50 years. And we talked to a bunch of VCs and we're just really looking for help on company building. Mm-hmm. And so we took the check and we put it in a savings account and never spent it. Yeah.
But my question is like, when you went to these investors and you said, hey, I'm going to leave this therapeutics company and start sequencing as a service, what were the types of things? Presumably they were like, oh, there's 1 million incumbents. You're competing with, this is kind of a commodity.
Yeah.
What did they say?
So we hadn't started the company, right? We were raising for that. And the issue is that if you're running a therapeutics company, you don't know if it's going to hit for like 5 years plus probably. And that for me, as somebody who wants to just be doing stuff, today, seeing tangible benefit today is not exciting for me.
But people would call this a boring company.
Yeah, everybody called it a boring company. Yeah, everybody said, my background is, I was working in a top synthetic biology group, doing, I think very good research. I had a science paper and a cell paper, a lot of options, and pretty much universally, all of my friends in this ecosystem who are, professors at top schools and even people that were running companies in this very city, they said functionally like, you're going to be throwing your career in the garbage to do this thing that like, yeah, you could probably make a couple dollars, but it's gonna be super boring and it's gonna be pretty capped on the upside. And like, you're not gonna be able to recruit fun people to work with and all these things, which has been, I would say, like universally not true. We have had a tremendous impact and, people want to work on stuff that has an impact. And so frankly, like we ended up being able to get some very, very amazing people to work with who didn't want to spend, 5 years on a problem that, may ultimately just, not be a viable drug for some reason.
And when you went to these investors, was it ever worrying that, now you kind of have protection, right? You have trade secrets, you have like things you've developed.
Yeah.
But at the time you were like, oh, we're just going to take this off the shelf technology.
Yeah.
Was that ever concerning to you? Like, did people express kind of concern that you didn't have your own tech?
And so there's a couple of questions wrapped up in that. One is like, what would investors think? And like, I don't know. I mean, I'm not too worried about what they think. Like you talk to enough of them and you can see the like herd mentality when you're talking to that sort of person where, they're not interested until everybody else is interested and then they're interested, which sort of tells you that like they're not independently thinking about this stuff.
I'm going to publish this on Twitter. You're going to upset a lot of people.
No, it's fine. I mean, like, I have a lot more I can say about this. I think they all recognize it. And it's just human nature, right? Like nobody is like confident. People are much more confident when everybody else is coming to the same conclusion, right? That's just like human nature. That's, I would say, like sound logical reasoning for a human. However, you know, when you're thinking about like building a business or really building anything, you can like read a book on how to do it, right? Like a get rich quick scheme on how to do this. Think about like drop shipping. Okay, you can get rich on drop shipping, and you can go read a book on how to do this, and you can be convinced that oh, you're going to make so much money as a drop shipper. And the question is like, has anybody ever made a lot of money on drop shipping? Yeah, I'm sure plenty of people did. And then they wrote the book, and they started posting about it, and then like the secret's out, and there's no money to be made there anymore. And this is what you see everywhere. That if you really want to have an outsized impact, it needs to be like an original thought, or at least a thought that you cribbed from somebody else that like has not escaped into the broader ecosystem yet. And that's sort of just like intrinsically sort of an isolated thought that you're having. And you have to convince yourself that for some reason you have secret knowledge that other people don't have. And if you know your personal shortcomings and your personal strengths, ideally, the thing that you have a lot of conviction around fits well with your strengths. And you start to say, hey, I might be the only person in the world who has thought this before or has been at the convergence of these things. And I can put this together in a way that other people, haven't seen. And I think, if you're rational enough, you can convince yourself that even if everybody else isn't seeing it, there's something there. And so that was what happened with this was, yeah, I was heavily doubting myself for the first week or something about if this was a good career choice. But
The first week,
maybe a week.
I doubt myself for years.
I mean, look, the thing I do, and I'm sure you do it, I'm sure most people do it, is I just start calling up all my friends and say, like, I've got this idea. What do you think about it? And, some people will say, ideally, your friends can give you honest feedback. And if their criticism makes logical sense to you, then that's a good thing too. You're very grateful to get that feedback. And if it doesn't make logical sense, then you're grateful to know that like all the objections to this are not valid. It's like when you're reading an Amazon review, right? And you see it's got a bunch of 1-star reviews, you go and you look at the 1-star reviews. And if the 1-star reviews are like, my package came damaged or whatever, you're like, okay, I'm not worried about this. But if it's like, a bunch of them say it stopped working after a week, you take that seriously. So it's like, you'll always get a lot of objections, and you need to be independently able to assess if those are valid or not.
When you do interviews, everybody always asks you about, growing the company to 40 whatever million annual revenue in 3 years for such a boring company when you're just using existing sequencing machines. And now all these investors are probably like, what is the next Plasmidsaurus for this or for that? And yet there's all, I guess what I'm asking is like, and yet there's all these incumbent CROs. So it's like, in what ways is Plasmidsaurus actually the first to do something? Like, people kind of have a feeling that you're doing things differently, but do you have like a very serious sort of thing in your head of like, this is why we're different from all the other CROs. This is what enabled us to scale so quickly. Like, is there actually like anything specific?
Yeah, I think there's a lot of coherent thoughts there. So our mission from the outset was very much around build things that will transform how people do their day-to-day research. And like, don't think too hard about monetization, like make sure that you'll be able to turn a profit on it. But just assume that if it's a thing that allows people to do stuff they otherwise wouldn't be able to do on a thing that they do a lot of, that there will be a very, like, you don't need to do too much market research to realize that's something worth doing.
But if they're not able to do it, how do they do a lot of it?
Well, so for people were sequencing a lot of plasmids, right? But they just weren't using Nanopore for that. And so the point is like, this is a thing that it would be better. Like new things were enabled when we launched whole plasmid sequencing. There was a number of new things that were launched. And it's sort of like, once you've done that, once you've got burned and realized, oh, I was being misled by the Sanger data in a way that was affecting my research, you're never going to use that again, right? So, and you can just look at what that market is and how many people are generating plasmids. And so, for a while, when I would think about this stuff, it's, I don't know if other people could have thought about doing the business that we did. I certainly have talked to a lot of people that say like, oh, I was going to do that. They didn't do it. But like, oh yeah, I was thinking about that. We focused I think a thing that differentiated us, and again, I don't really think of us as a CRO. I think of us as a service or a products company. But I was very focused on offloading as much mental burden from scientists as possible, right? So like a lot of times people say, oh, I got this technology and get out there and people will use it. We were very much thinking like, um, somebody's going to put this in. They don't want to think about anything we're doing. They shouldn't ever even care what technology we're using. They're just going to know they're getting back this data in a way that doesn't even require any additional analysis. They just get a check mark. This is, these ones are right. And all of the data is there. It's all, if they want to dive in deeper, we have, I think, the best analysis suite as part of that people can get into, but they don't have to. And nobody else has like used that mindset. Nobody else has. The thing that I feel very proud of is, we built this plasmid sequencing company. It was very popular. And it's like, could somebody else have done it? Yeah, somebody else, if somebody else did the exact same thing as me when I did it, they could have done it as well. But then we launched this product last year, this RNA-seq product, which RNA-seq is where, you're just you're counting the RNAs in different samples in different treatment groups, and you're looking at differences in those between treatment groups to learn what the biological differences are between the samples. Okay. And RNA-seq was first published, I think, back in like 2009. I think it was like Barbara Wold's group at Caltech was the first one to do it. But this has been around for a long time. And until we launched our product last year, there was no easy way for you to just quickly get this data from your biological samples. And so that tech was functionally sitting there for over 15 years for somebody to commercialize it. And people had done RNA-seq services, but it's a thing where you're gonna, you got to prepare your samples all in a laborious way, ship them off. You got to wait 4 to 6 weeks. Then you get back a bunch of data that you have to analyze. And we were thinking, okay, people just want to know what's the difference between these samples. So they just submit us their whole cells, in preservative. And then 3 days later, they get back the wholly analyzed data. And this is a hard thing. Like, my team worked really hard to make this possible. But it was totally achievable. And I don't nobody's going to tell me, oh, I was thinking about doing that. I've actually talked to some people at large services companies who told me, like a CEO of a large service company said, we tried to do that and we could not execute on it at our company. And they're a good company.
Why not?
It's just hard. It's just hard.
But what is hard about it?
Every single step is hard. So imagining the concept that you need as raw a sample as possible for people they just want to drop it off. Having very strong logistics to get that into your lab as fast as humanly possible, having robust, arbitrarily scalable infrastructure that allows you to extract things at high scale with high quality is a thing that I think we do better than anybody else in the world. Getting all of your machines online. So, for our Oxford Nanopore sequencers, for our Illumina sequencers, we are heavily invested in being able to control those things remotely and get all of those readouts in real time so we can be making real decisions in a way that's like a ton of engineering work by engineers that are of a higher quality than are any of these other companies. And they were working very long hours to get these things up and running so they can do this, to automatically kicking off the bioinformatics workflows in a way that we can be confident is giving people back accurate data. I mean, everybody else was just still in the mode of actually running individual scripts on each thing and looking at the data as it comes off, and us just saying, no, we need a scalable solution for this. And then putting the effort into the UI. So again, they don't need to know anything about the sequencing technology. They can just literally type in their gene name, or actually right now you can use a language model to say, I want to see all the genes involved in like the HIF pathway or the BMP pathway. All those plots automatically come up and show you your differences. There's just, I don't think there are any other companies that are able to execute vertically at such a high quality across the board.
And do you think RNA-seq will be your biggest revenue maker compared to whole plasmid sequencing? It feels like when I was an undergrad, it took me 6 months to do an RNA-seq experiment. And it was, as you described.
Yes.
And also, again, you chose a price that is like, you could charge so much
more. It
more.
was faster. It had more, the analysis was all better. It didn't require them to do any of the prep work they would otherwise have to do. And is a lot faster. And then we underpriced it compared to anything else on the market. And, our take is like, this is a technology, so just getting back earlier, something that, everybody can do. Anybody who's doing any tissue culture work or even, organismal work, they are looking for changes in gene expression. So that's the market, right? And we just had to convince them that this is going to be easier and higher quality than what they're doing. And the way you do that is you get them to try it. And then, when people, you ask them if they'll say, oh, no, I can do that myself. I'm good at that. And I do this. And then once they try it and it's better than what they would have done themselves with less work,
And it's
they're
$80.
converted. $80 for commercial. It's $50 for academics, which is a lot of I mean, but the thing is, like, well, first of all, academics are in a tough place right now, monetarily. But also, like, that is where it matters, right? Like, if you're charging, we could have charged $150 for this product. I'm sure we would have had very good volume at $150 because that's competitive with everything else, and it's better on all these other axes. But the adoption people would start to say, oh, like, I don't want to send this order because, like, it's gonna be too expensive. Now they can send, you would think if it's a third the price, they could send 3 times more stuff, but they're actually going to send even more than that because it's a low enough price that they could do it for functionally every sample that they would otherwise be doing qPCR on.
But so the immediate response suggests that it will be even bigger.
It will definitely be a larger, yes, it'll be a bigger product than whole plasmid sequencing. It's already getting close, and we've only scratched the surface on the market. But I mean, the goal, like, how do we continue to grow? You have to keep thinking, what are these products that that'll just be a thing where once people use them, they're like, how did I ever do my research without this?
Will Plasmidsaurus always be a sequencing company? Or is there a future in which you also do Western blots and, just other things much cheaper than competitors?
Yeah. I will tell you, over lunch, it's now like 2 p.m. or something like that. Over lunch, I got an email from a customer who was like, you guys really need to do mass spec. It's exactly the perfect use case for you. It's scalable in all the right ways. Everybody needs it, and it's hard to use right now. Will we offer mass spec? I'm not promising you we will offer mass spec, but like that sort of thing is very aligned. I think we're in a weird place right now with sort of biology and how everything's going. So the scientific thinking is starting to be more and more levered by the frontier labs. I mean, there's many non-frontier labs who are trying to help this, but clearly we're going to a world where scientific thinking is heavily going through Anthropic, OpenAI, and Google. Clearly that's where we're going, and there's no sign of things slowing down. As we're talking, GPT 5.5 was just released, and it is a dramatic improvement over other models on this axis. And it's just going to continue. The bottleneck for scientific progress, and that's really what drives us, is how can we get like new human therapies out faster? The bottleneck is turning, scientific hypotheses into data, right? And data is kind of like, I don't know, it's not my favorite word, but it's, stuff that you can reason over, that a language model can reason over. And the richest data is sequencing data, but there are many other things as well where the bottleneck to scientific progress is implementing those things experimentally and getting data out the back end. And we will be operating in many places in addition to sequencing.
But you told me before this call that you're not selling data to AI labs.
Yeah.
And yet, if all the reasoning goes through Anthropic and whatever, then at some point you have to sell.
Well, we don't sell it to them. I mean, so like the thing is, like, think of us like Dropbox, right? Like, Dropbox is not selling your personal data to other companies, right? Or Google Drive or whatever. You are assuming that stuff, and the terms of service, I assume, mean that, they're hosting your data, but it's your private data. And, it's important to us that our customers feel the same. And we've never sold any of our customer data to anything else, and we wouldn't do that. A frontier lab can pay us to sequence stuff for them. And, many biotech companies right now are paying us to train their models, to generate data to train their models. Very excited to do that. But, people's data is their data. It's not ours to be training on. We're helping them out in that way. And yeah, I don't think it's our place to sell the data. People would like to, we talk to investors who are very excited about the idea that, oh, we have more data on RNA-seq than anybody else. We have more data on plasmid. We have more data in many other ways than anybody else. But yeah, we don't sell that. It's not ours to sell.
But are you explicitly looking at the market and seeing where the foundation model labs are trying to collect data? Like single cell RNA-seq might be a really good
Perturb-seq
place.
is a big one.
Yeah, Perturb-seq to collect.
Yeah.
For virtual cell training.
Yeah.
Like is Plasmidsaurus planning to expand into that?
You can imagine anywhere where there's a lot of data being created. If we feel like we can provide value there that isn't currently be provided, it's a thing we're seriously looking at. We're very aware of the fact that the more data that is digitized, the better it is for science. And one of the things that starts to be, so if you take a step back, you start thinking like, there's a lot of talk right now about lab automation. And is this the solution for whatever America's biotech woes, or in general for biotech progress? I don't think like automation per se, we don't just need to have a bunch of like, dark labs where they're just running autonomously. But having all of the information be as machine readable as possible is useful. And so like places this can show up, like right now, one of the core things you do, or I shouldn't say right now, for the past, 80 years probably, one of the core things you do as a biologist is you run gels. Okay. So you asked earlier about Western blots, but like an agarose gel is sort of a core unit of bioresearch. So you have some DNA sample, you've maybe done PCR, you've done a restriction digest, you're going to electrophoresis it through this algae matrix called agarose, and then you're going to stain it with a UV fluorescent dye, and then look at it and see how big your DNA is. There's like a core thing that people do. And that doesn't work super well with automation. Okay, like that's not super machine readable. There's a lot of things that don't work about it. But you can very easily do the same thing through capillary gel electrophoresis. And there's like a thing called a tape station. There's a number of devices that do this, and you get functionally the same readout. And if you, instead of running an agarose gel in that way, you run something through an automated electrophoresis system, you get the same readout, but now it's immediately readable into the machines. And I think things will probably start pivoting that way like pretty quickly. And again, we are very excited to be able to help people do that. And the more that stuff gets in the loop, the easier it is for scientists to reason on their data and to make scientific conclusions using language models or just because they're really smart. And but anything we can be doing to help accelerate that, we're very excited to help out with.
To kind of push back on what you said of, oh, we're not going to sell data.
Yeah.
If you were serious, customers wouldn't like it, presumably, but if you were serious about speeding up scientific progress, why not just sign a partnership with Anthropic, or like, wouldn't that speed up progress much faster than saying, hey, you have to pay us to do all your sequencing runs, but you don't get access to this huge moat that we've already built?
Yeah. Well, if you look at like the frontier labs, like sure, I'm sure they'd be happy to be training models on data like that. But I think a lot of what they want to do is to enable, rather than like training models on the user data directly, they want to enable the researchers to use their models to do their research. And they're very excited. I mean, you can see this, both Anthropic and OpenAI have big bio programs that they're, OpenAI announced this Rosalind model last week. Anthropic, is vocal about their interest in bio. And you can certainly imagine partnerships where we make it easy for customers to get their data into those systems. And we, as part of our RNA-seq pipeline, we have a way where you can be interacting with it via Claude directly on the results page, pulling in all of the raw data, doing analysis like that. But I don't think, like the pharma companies, and all they want to own their data. They're paying a lot of money to do that. I don't. If they want to have a partnership with Anthropic or somebody to facilitate that, we're happy to be a party to that, but it's not really our place to sell it.
It's interesting because, one thing that I've been thinking about is when you launched, you didn't have any IP per se. But since then, you've amassed all this data that's, interesting, proprietary. You can train better predictive models to improve the accuracy of the technology. But also, it seems like increasingly your moat will just be the way that you analyze data, right? Because Nanopore and the technology developers never really build the infrastructure to help users analyze their data.
Yeah.
But Plasmidsaurus has built these standardized bioinformatics pipelines.
Yeah.
And so presumably, like what you're saying, if you get enough data all in the same format that has all been kind of processed in the same way, that is the value.
There's a lot of value there. There certainly is a lot of value there. And one of our moats is that we have the best user experience for people that want to analyze. So we have standardized bioinformatics analysis, but also the UI is very differentiated. I don't know if you've seen it, but it is very differentiated for all of our services. But I think, we have been intentionally building that out for the past couple of years as a moat. But that is where language models are strong, is on, maybe you don't want to actually go to our website. You just want to ask, Claude or ChatGPT to look at your data and present and make the plots for you. And they can do that now. I mean, I would not trust it for publishing without combing through it. But, those models are only getting better. I think our core moat is that I think we have the best ability in the world to take raw biological sample and convert them into high quality data. Like, we have very strong automation. I think it's differentiated in many ways. We can scale that in ways that other people cannot, with a speed that other people cannot. And we can adapt it to arbitrary new standardized workflows because it's all in-house. We don't use a third party to build out our automations, stuff. So to me, that's when I think, like, what's the real core value? It's that there is nobody else who is better positioned to digitize experiments than we are. And so right now, again, that is sequencing, but you can imagine a world where you're using a language model to make a bunch of antibody variants, make 10,000 antibody variants, and you want to see, what is the relative binding strengths of all those or whatever. And that would involve getting those oligos synthesized, getting them into cells, and then getting a readout, which could be sequencing, probably would be sequencing. And I think we are well positioned to do that.
And so if tomorrow Nanopore said, we're not going to sell our devices to Plasmidsaurus anymore, and we're going to compete on sequencing as a service, would that not be concerning to you?
Well, it would be in violation of our agreements with them, first of all. So I'm feeling confident on that, because we do have partnerships with these companies that we work with.
But this seems to be a common mistake amongst people who develop technologies, don't then sell access to the technology as a service. Like, Natera is worth more as a company than Illumina.
Yeah.
Even though they just use Illumina machines to sequence like embryos during IVF.
Yeah.
And so Illumina presumably looks at Natera and is like, whoa, why didn't we just do that with our own technology?
So it's interesting. Yeah. I mean, without getting into specifics of any of the companies, there's a rich history here. So, all of these companies, when you're building an enabling technology, I assume the scientist, when he's, coming up with the idea, or she is coming up with the idea, they're thinking they'll get like so much of the value that's created on their devices that they're creating. And it's a difficult line to walk, because you have to have the entire world excited to develop on your platform to push it forward. And so if you are too intentional about saying, hey, we're just going to do everything, you will not get the adoption that's needed for your company to be successful. And so Illumina was actually quite concerned early on about this. And, ultimately a number of people developed kits that were Illumina compatible that my my guess is Illumina was not actually excited about that because they wanted to be selling the kits. And I think they ended up buying Nextera, for instance, but, and trying to do diagnostics. But, you know, the way the economy works, right, is for these things to be successful long-term, you really have to open it up and allow everybody to try and build on your platform. And so like Illumina, so why does Natera have the valuation they have? And I think Illumina has higher revenue and probably higher profit than Natera right now. I'm not sure. So that part of that is just valuation. But, Illumina would have never, if Illumina in 2008 or 2009 was like, oh, we're going to be doing diagnostics, there's almost 0% chance they would have had Natera's MRD product. They would have ever developed that, right? It would just required
MRD.
Minimal residual disease. So the way Natera works is interesting. Natera was started as a pregnancy diagnostics test to try and see, does your kid have trisomy 21 or whatever, by just doing a blood test. But the reason they have such a high valuation right now is it's for cancer screening. So if you have a cancer mutation, so you have a cancer, in that cancer there is a mutation. And what you want to see is over time, is that mutation increasing in frequency in your blood? And if so, it means that your tumor is growing. And the way you actually do it is you biopsy the tumor, you deep sequence the whole tumor, you look for mutations that are specific for the tumor, then you go back and you sequence the person like every couple of months or something like that, and see, does that specific mutation changing in abundance over time? That's what their product is. So it's minimal residual disease, the residual disease being the cancer. Illumina almost certainly would have never developed that specific product, given, certainly wouldn't have developed it by now. So, this is just how the world works, unfortunately. People, you kind of hope that companies will build on top of you and have a higher valuation than you do, because that means you're being successful.
There's a testimonial on your website from someone who said that their sample arrived at Plasmidsaurus at about 8 a.m. on a Saturday, and they had results at 8:50 a.m.
Yeah.
Is that real?
And they dropped it off the previous day at like 5 p.m., right? So,
But is the turnaround
real?
actually 50 minutes?
So when we're thinking about plasmid sequencing, and this was a plasmid, I think we can return data to customers. That's about the fastest we can return data to customers. If we are like, everything is going smoothly, right? Your sample gets received and is immediately processed, as the first sample and all these things. I would say that's about right. And the way we can do that, just as an example, and we're the only ones who can do this in the world, is, as I was saying earlier, Oxford Nanopore gives you data in real time as you're sequencing. You don't need to wait till the run's completed. So as we're sequencing it, we can start looking at the data and say every 5 minutes, do we have enough data here to return? Like do we have enough data that if we sequence deeper, it's not going to change their results? And then as we get enough data per sample, we can start emailing people their results, while other people who were sequencing at the same time might still be a couple hours because, their data is of lower quality or something like that. So that's one thing. That's one way we're able to do it very fast. We are hyper optimized on speed all throughout the company. This is like our core value, or a core value of us. So that means that every step in the process has very intentionally said, what happens if we cut this shorter? What happens if we cut this shorter? Are there things we can do to eliminate steps so that it'll be faster? And this is from opening the packages functionally to getting them into our database as they're being received. We use custom hardware for all of that. The automation equipment is all heavily customized. We have 3D printers in every lab so that we can customize labware for everything. So yeah, I would say that's about as fast as is possible.
So we had dinner recently, and you told me about your visit to a USPS shipping factory. What was that like?
It was UPS shipping factory. Yeah, it's incredible. Like when you think about all these packages are being shipped around the globe, for all of these major transport carriers. uh, and they're magically appearing at your door very quickly. And, there is a ton of people whose entire lives are dedicated towards making that as seamless as possible. And a thing you don't think about except for when your package is delayed. And, you see like the Amazon thing that's like, I don't know if you've ever gotten this, I've got a couple times. It's like package delayed, train derailment. And you're like, what? Train derailment? Like what? But like, I guess it's a thing. The way the air shipping works is they have all these, jumbo jets, and you get to go inside them. And inside the jet is just the entire fuselage is filled with wheels on the bottom. And then inside of the plane, which is a tube, right? You got to fit all of the packages somehow in a tube. And they've gone through and they have custom boxes that are shaped so that when like 10 of them are put together, they form a circle, and then, that goes in. So there's, ones that have all these weird shapes. And they show up to the facility. They just start unloading them. The wheels turn on and just squirting them out the back. And they take them and they just feed them into one side of this giant logistics, like, 15-story logistics facility where the packages just go in. They are having scanners everywhere, scans them, and just as little hands, levers sorting them all. And then they come out the other side. And, each door is a different plane it's going to go onto. And then in whatever is like an hour from when all of these planes land, come out their side and then load them back up and just, take off one after another. And I think it's like it's extremely impressive logistically how they have simplified it so much, and made it straightforward. We were there because we work very closely with UPS. And so one of our core, so I talked earlier about how one of our core competencies is speed. And that is something that is front and center of every decision we make as a company. And that enables us being by far the fastest way to get your sequencing data back. But it also is like a philosophy that we have, at the company is internal processes is all about speed, right? So like we decide we want to do a new product, right? How can we do that as fast as humanly possible? How can we de-risk it as fast as humanly possible? How can we get a prototype out to customers as fast as humanly possible? And it's core to everything we do. When we started in my garage, it was just me and my, couple co-founders. And even then it was, every decision was like, when we have a network of labs around the world, how are these things going to arbitrarily scale to be fast in all of those situations where, somebody doesn't have my tacit knowledge? But anyway, I was at UPS because we have a close partnership with them. And we actually, one of our facilities is located in Kentucky, at Louisville, Kentucky, where the UPS national hub is. So we can get packages into our lab very rapidly without needing to do that second air transport trip.
And when you were expanding the company, how much time did you actually spend, studying logistics maps and figuring out where to position hubs strategically? Like, was that much harder to figure out than you thought, or did you just hire somebody who kind of had already done this at a different company? Like, how much of that is sort of trade secret?
Yeah, I would say like when you're starting a company, like we did, which is, bootstrapping it,
Mm.
pretty much everything you're doing, every new important aspect of the company, you're intimately involved with. And you can't outsource it to somebody else. You can talk to people, but nobody is going to care as much as you do, right? And so you can probably go about 3 times faster, I would say, if you do it yourself, because you're working like 18-hour days, um, maybe more than 3 times faster. But you're working like 18-hour days and you care a lot and you are very motivated to make as effective progress as possible compared to somebody who's just trying to collect their paycheck, right? And make sure that they're going to get paid for their consulting gig. So we did all ourselves. The first, and like everything else we do, the way you do it is one step at a time, take the plunge and then try and fortify from there. So the first real logistics thing we worked in, and we intentionally wanted to get hubs in all of the big biotech cities. So that's, Boston, San Diego, San Francisco, and we were in LA because I started out Caltech, it's in Pasadena. And we we got a courier that was driving up every day from San Diego. That was easy because we could keep running that out of our lab. And then our plan was to open our next lab in Boston. No, our plan was to open our next lab in San Francisco because it's like a 1-hour flight. So if something went askew, I could fly up there real quick and help them sort it out. And so we started thinking about opening in San Francisco. And then instead we're like, oh, we don't need to actually open a lab in San Francisco. We can just solve this logistically. So got out the, phone book or whatever, got on Google, found couriers in San Francisco, started telling them what we wanted to do, which was we wanted them to drive around to all of our sites in San Francisco. Then at the end of the day, drive them to the airport, put them on a cargo flight, fly it to Burbank, and then have a courier go from Burbank to our lab in LA. And so we started doing that. We ended up opening a lab in San Francisco eventually, but for over a year, every single day, one of us it was actually Kevin, my co-founder, was the one who was most involved in this, but it was look at the flights, see, is anything delayed? If not, get stuff on there. If it is, figure out how to reroute it. And, at some point you build out systems that are efficient at this, but it's a lot of work. But then you learn a lot of, by being involved in the process, you learn all of the little tricks. And I remember there was a day early on. So one of the things about flying out of San Francisco was that depending on the month, we would have to fly out of San Jose or SFO or Oakland because the way you ship these is on commercial airliners, but they have like a cargo section of the airliner that you can do this with. And they would change the flights like randomly without warning. So it's like, you're taking the 6 p.m. flight every day. And then one day there's no more 6 p.m. flight out of that. And you find out like that day or like the day before. And so then it's like, okay, now stuff's going out of Oakland or now stuff's going out of San Jose or whatever it is, and being agile with that. But I remember there was a day where we were shipping from Berkeley to SFO and there was a hijacking on the Bay Bridge. And so we missed our shipment. And it's so important for us to, get people, people have to know they can rely on you to get their data. And once you start breaking that trust, they're no longer they're going to be second guessing every time they use you. So there was the hijacking, it got delayed, got to SFO, but there's a flight curfew in Burbank. So you can't land flights there after 10 p.m. So we had to reroute that to LAX, which is a much longer, it's like an hour drive, over an hour drive from our lab. It ended up landing, at midnight, but logistics is slower there. So the package wasn't released until like 1:30 a.m. Drove out there, pick up the package, and it was me, waiting in the parking lot. Package is released, drive it back to the lab, get into the lab at like 2:30, have my team there ready to start processing the samples when they get into the lab, use everything to make it go as fast as humanly possible. And then customers still got their data on time, right? And they have no, we didn't send an email saying, sorry, there's hijacking, you didn't get your stuff. It's just, business as usual. So that was a learning experience. And then as you continue to grow, we now have couriers. Either some people work as contractors for us, and some people it's courier systems. But we're probably operating in like 50 cities or something like that, where we've got people doing stuff for us.
How many customers do you have in China?
We have customers in China. We do not have a lot of customers. So most of our customers, we have intentionally not entered China as a market because there's so much opportunity elsewhere, and it's just difficult to operate a U.S. company in China. We do have customers there, and they're typically people who were using us here and know that our service is so much better that we have boxes there, but we have not ever intentionally grown in China.
You could, I think I saw on your website that you have, what is it, 70,000 scientists or something? That's publicly on your
Yeah,
website.
we have more than 70,000, yeah.
But just in the US, there's like, I don't know, 200,000 people that work in pharma, 100,000, 150,000 biomedical scientists, I think.
There's over 1 million people that can be using us around the globe.
Around the globe.
Yeah.
So the market is presumably huge in China.
Very big. Yeah.
Why not, why not actually make that an effort? Are there more competitors in China?
Chinese people are, as we're seeing in the biotech industry broadly right now, they're very interested in in-housing solutions that are purely Chinese. We have done very well at providing impeccable service globally. I think in China, the local competition is very strong. And it's just, relative to the rest of the world, it's not a good use of our effort.
If you had to give a new founder, I think you've commented on some of this, but if you had to give a researcher or founder a playbook for finding the next Plasmidsaurus type of thing, what would the steps be? What advice would you give them?
And, the core thing is you just have to provide immense value, right? Like that is the core thing. And it has to be like a big market, right? And it's hard to find things. I think if you're just like, I'm going to go on a walk and come up with a business idea, it's really hard to find something that nobody else has done. Something that was helpful for us as the founders were all deep in research. And so, we understood the things that seemed dumb about the way the world worked. And we're like, it seems like there's a better way to do this. But somehow I think we're very fortunate that it fits our personalities well to address those needs. Cause I talked to plenty of other people who are smart, and when I'm workshopping when they're workshopping ideas with me, a lot of times they seem like inherently like it's not going to work, like because just market sizing or positioning. I think the core thing for me is like do it as fast as possible. Like get your idea out there as quickly as possible and iterate on it with market feedback as quickly as possible. Like, do not spend a year ruminating on how it's going to be perfect, because you will certainly end up in a work. If you spend a year thinking about how to make the perfect product, or you spend a year getting something crappy out there and iterating on it for a year, you're going to have a way, way better product if you've been iterating on a year versus thinking about it. So I would say the core thing I would always say to somebody is they're like, oh, I'm just going to read this book first, or I'm going to take this course, or I'm going to do this, do that, and then I'm going to start a company. It's like, just go try and provide something of value as fast as humanly possible.
Yeah.
That'd be my general core advice.
And what would you be working on if Plasmidsaurus didn't exist?
Well, there's the hypothetical. If I had not started Plasmidsaurus, I would have almost certainly started a therapeutics company. And then it probably would have gotten acquired into some pharma company or something like that. And that's what I'd be doing. If I if I fired myself and I was no longer able to work at Plasmidsaurus, I would, there's just so much interesting stuff. I'm like the kind of person who's interested in everything. So there's so much interesting stuff to work on.
That's my problem too.
But I think, what's going on with language models right now with AI is certainly would be an exciting thing to be a part of. So I'd probably be, working there, would be my guess.
But in biology,
To enable biology from within them. But there's just clearly so much value to be added there.
what do you think are the most promising things being done with language models for biology? Would you go to Anthropic or would you go to Edison? Maybe you don't want to talk about specific companies.
Yeah, I don't want to talk about specific companies, but it seems pretty obvious to me that the frontier labs are going to win long term. And just to explicitly, this is Anthropic, OpenAI, and Google
On scientific reasoning.
on everything, including scientific, on reasoning, right? On reasoning. And that's certainly, I think, would be most appealing is to help build those.
But so what actually has to be done there? What are
So, okay, the way I think about how this is all working is there's the five steps to scientific research as I think about it, which is which kind of mirror a paper, actually, as I articulated. But first one is figure out what you want to do, which is you maybe know two technologies that you think you could put together in a novel way that are going to create something which isn't otherwise possible. So that's step one, figure out what you want to do. Step 2 is like come up with your experimental protocol for how you're going to test it, right? And so this is going to involve reading a bunch of papers and saying like, how do they do this thing? And like, how can I borrow these things? And what am I going to do? Step 3 is like doing the hard wet lab work. So actually following out the protocol, doing all the measurement. Step 4 is analysis of your data. So it's like bioinformatics or whatever you want to do. And then the 5th step is like determining, like, did it matter? Did you learn anything? Was it useful? Is there, if you're, is there a drug that can target the thing you did or whatever? Like trying to put it in context, in the greater scientific context. So there's those 5 things. And I think 4 of those things, all of them except for the wet lab biology, are massively levered with language models. So identifying if a problem is worth working on to begin with, play with ChatGPT for 5 minutes and you'll be getting great insight into how novel this is. Coming up with experimental protocols, like, historically you would spend, if you're doing good work, you're going to spend maybe a couple of weeks poring through papers trying to figure out what the best possible way to do this is. Which method should you be using? Which buffers should you be using? Blah, 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 blah. That is massively, massively accelerated with language models. It's the thing right now where you can just ask it what to do and follow that protocol, and you're probably in a good spot. The bioinformatics analysis, obviously massively levered, like so much easier to do any sort of analysis right now. That's the kind of the sweet spot.
Wait, so you're saying you would do that instead of wet lab stuff? Even though
I'm
wet lab
saying
stuff is much
all
harder.
of those things are accelerating biology in a major way. And so like the thing that drives, like the core thing that I'm interested in is how can we improve human health as quickly as possible, right? So my mother recently passed away from a disease that is not treatable with current methods. And she was very like, oh, is AI gonna, because you hear all the news all the time about, oh, AI is gonna solve all these diseases, blah, 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 blah. And so she's like, is it gonna solve it? Is it gonna solve it? And I sort of had to like, it's like, this is hype, right? But I am like very optimistic that if we look back, say 10 years from now, somebody who has that same diagnosis is going to be in a much better spot. And I think that is all massively accelerated by the work that is happening, I think AI, language models, will be accelerating all of that dramatically.
Well, Mark, thank you so much for coming on the podcast.
Thank you for inviting me. This has been wonderful.